I heard the weather's always as beautiful as it is today. We had a nice hike. Yeah, it's really great fun to see so many of the single cell friends and colleagues here and what's going on. And there was a bit of a danger sometimes getting a bit too like into the early days of what happened in single cell biology and how this has been blowing up. But I just want to share a few new developments we've been doing in the lab and sort of maybe also what the field has been doing and how we attach to that. I'd be very happy to be interrupted in between. Since COVID, at least my attention span for 45 minutes just listening is not there. So I'm totally fine if you want to interrupt me, just go ahead. So what I want to talk about a bit is sort of first maybe sort of the big picture, right? I think in the sense, this was, you guys, does someone still remember from the lifetime proposal? We talked about how to set up something on the European level. This was one of the efforts at the time. I think the sketch and concept is still kind of cool. So where, you sort of had cells sort of maybe living happily their little life, and then it sort of maybe turn off and do all these bad things. And I think it's sort of a nice machine learning question to, let's say, maybe learn this cube to some extent, like in a data-driven fashion, just draw it. But then also, and I think this is important for sort of maybe building even a scientific theory of cell behavior, maybe how to be able to systematically act upon. Okay, so knowing when you stand here, I need to do ABC to go in this direction. Let's say I'm here, I want to go here. What do I need to do actually to sort of go around? So this perturbation prediction, I think, is a quite exciting direction. So I think on a super simple thing, what we kind of want to do, we want to do some type of representation learning of what this state space is, maybe according to different data sets, maybe also fill up one modality that we don't have, but then sort of connect it in a dynamic fashion. That's, of course, what sort of attaches nicely to what Bart is doing here, maybe actually really measuring time things. I did come into the field from time-lapse microscopy. So at some point I had some dynamic information, and now we just have a sort of lot of samples and we can reconstruct these trajectories, right? So basically writing and maybe up some type of system like that really tells us when we are here, what would be sort of the next state, and maybe also distribution of next states and so on, right? But then of course, like maybe if you really then see when some of these branch things happen, can we zoom in here? And can we learn what type of change we need to do, what type of perturbation to then sort of move on to this? Yeah, you could maybe call this full causal reasoning. I have a student like every 2 or 3 years trying to do like the full causal setup, and never adds so much. So we then go back to the more sort of predictive type of setups. But I think that's sort of maybe a bit of the holy grail. So we've been doing a lot of engineering things. I just wanted to put one slide up at the beginning to say that even though there's maybe always some fancy, nice machinery that you can do, in practice, a lot of that is just robust engineering. So, first thing is an analysis in my lab took 1.5 years. We said we need to do a bit of a better system. We set up this software that now we turned sort of, I think, a very nice international consortium of people called SCVerse, with a lot of colleagues and friends from Olli Stegle, Francesca, Dana, Aviv, Sarah, and so on, where we now have a shared data structure, particularly in the Python world, how we sort of share single cell plus multimodal versions, also spatial, a sort of core analysis package that's sort of jointly maintained, and that's sort of a way how we keep this up to date. So if someone's interested in maybe hopefully using, many of you do, but maybe also sort of contributing to that, I think it really helps sort of maybe make some of the analysis that it just sort of maybe sort of just spottily happening, maybe joined together. And we talked about benchmarking. This is maybe also something that can feed in there. We have similar things for the spatial side. This one was got quite popular some time ago, sort of just spatial neighborhood analysis and so on. And together with Olli and Josh, we've been coming up with this data format that sort of a multimodal fashion allows you to just stack together spatial, different type of sort of multimodal observations, jointly transform and operate on top. Whereas, in this world, stuff is kind of a bit more organized. Here, there's so many different formats. So if you can even come up with one way how we store these things, we're building a database for that as well. I think for training and some of the large scale efforts that we, Charlotte and other labs, sort of go for, I think we could really be profiting from this being robustly there. And now, quite recently, we've been sort of coming into the idea of how to do many perturbations at the same time, came up with a setup for that as well. All right, and maybe the last thing I wanted to say on the community side is, of course, like in practice, you mightn't even care about some of these detailed tools, and there's like too many around anyway. So what should we then actually go for? There's like solution from colleagues in French, such as Raoul's, who has like this sort of monolithic framework like Surat, where have each of these steps some sort of proven best practice. We maybe sort of have a more modular approach where people can do different things, and we sort of write about it. This is this living website, single cell best practices that I also encourage you to take a look and contribute if you're interested. We're doing a spatial extension of this at the moment, so someone interested, let us know about this. So maybe sort of just go back to this thing we want to learn representations in this sort of flowy space, right? Very basic. I guess what you can do is you take these autoencoder type of settings. So you take a gene expression matrix, squeeze them down, blow them up again. You don't have labels, so you just make them as similar as possible to the input. So just a nonlinear PCA essentially. Then you clean up stuff, right? And that was sort of the first motivation of early papers. This was my lab in parallel to Neil Joseph's lab, sort of introducing these things, and it was kind of nice. But for me, the breakthrough thing was that this latent space actually has sort of some structure. You know, in this latent space, we get some much nicer information and things in there, and we see these transitions and so on. Since then, you can add all kind of constraints on top of it. But I think that's been interesting. So what do you do with that? Well, one thing, let's say we have a whole bunch of these data sets early on, where there were, I think, like 20 papers. Hey, we have built a lung cell atlas, and it was like everyone had like 3 patients or healthy subjects, did a bit of RNA-seq, which was sort of time very expensive, and they all didn't fit together. So we can inform these models, and sort of one very naive thing here is called a conditional autoencoder. Tell them about the data set as a label. Now you can use that information to sort of more efficiently encode here. So if this one is always sort of +2 versus this one, you know, it sort of can learn it here, and thereby moves those data sets on top of each other in the bottleneck. So it's an automated data integration type of technique. And that's sort of at the core of many of the downstream things that are maybe not transformer-based, but pretty much do the same. So this one, you know, you can sort of put together, and that's why, you know, haha, I hope you saw my nice animation. These are efforts I do in the morning. So you can now build this integrated atlas of things. And the human cell atlas, the first one we did, this was work by Lisa, PhD, and now just moved on to industry, and Malte, who turned to PI, where we've been building an integrated atlas for the lung in a really large-scale international consortium, the Human Lung Cell Atlas Network, where initially we took a bunch of data sets, harmonized annotation, metadata, and location, and then just put all of this together. And this sort of gives you then this map or this globe or atlas of things where you can then zoom into these continents and then get all of these different annotations. So basically, here you superimpose these samples across those very heterogeneous data sets, then you can jointly annotate. And this annotation is then something that, you know, people really use to then sort of map the new disease data set on top. So what's the finding? I think an early critique, we often get like, you know, I mean, I knew that, you know, in lung I had 82 cells or whatever, right? So of course you get some finer grained clusters, but, you know, one thing, for example, we saw here was one cluster that was in monocyte-derived macrophages. It was high in this one factor called SMP1. That's sort of associated with some cytoskeletal thing or scarring or so. And we saw that this was shared across a whole bunch of diseases that you initially wouldn't put together. So there seems to be potentially, there could be like a potential shared process, and, you know, if you want to do some type of targeting or so, that could be something to look for. So people got interested in this atlasing. Now we're doing this in the human cell atlas across pretty much all the different organs that you can come up with. We as a sort of as a network, as a consortium, here's some best practices how to do this, but I think it's really useful to essentially talk about these single cell data sets after harmonization in such a way that we find groups that we can give names to. That's what we humans need to talk about these cell types. And this is, I think, very, very, very common problem. Here's another example. This was worked together with Barbara Treutlein and Greg Camp's labs at ETH, your partner institution. I heard very easy to interact with her. So this is called the Human Cell Atlas for the organoids. And I think, for me, organoids is such a nice setting because it's not just cell lines. So, you know, variation there actually is kind of cool if you do perturbations, but you still can do them at scale to some extent. And Barbara's lab particularly has been scaling these things up. And what we did there was similar things. So there were, I think, 20 different papers or so that sort of have all different ways how to cook your organ. Okay, so these experimental protocols that, for me, always is a bit of a random local optimization of protocols to learn, you know, at some point these work. But why do you actually wait so long? Why you add this morphogen and not this one? You know, I mean, there's some reasoning, but, you know, there's different ones doing different things. There's some guided ones, unguided, but you can do again an integration. You write this conditional autoencoder type of setup, and then you get this map where all of them across different types fit into one thing, and you see there's some sort of developmental happening. There's sort of, you get the different cell types, and, you know, you can annotate and you look what's happening. So that's useful, and we describe this quite a bit. But what's now nice is because you have this integrated map, you can now ask, hey, I mean, in the end, I don't care about organoids, right? I care about the human system. There was now this paper from Stan's place, who actually did early brain fetal development, like first trimester or so. And you can ask, like, how similar is this to actionable development? Because, you know, these are essentially just some coordinates. You can actually try to throw them together with these sort of type of maps. And this is what we did here. And you get this map here, we essentially project this one into the human development one. The color now indicates whether, you know, this one, blue means sort of one of the organoid systems is actually being found in the human case, and red means no, these things are not found at all. So you see some things actually overlap nicely, but there's a bunch of things such as, you know, these ones here, for example, that you don't find in the organoid setting at all. And why? Well, because these would be immune-related things that, you know, the organoids don't have any vascularization, this stuff is genetic. And then there's a whole bunch of other things that you can quantify. So if you just put the max presence score here, then you see a bunch of clusters here that don't match up. And there's, for example, a few here in thalamus, midbrain, and so on. And now one question you could ask. I remember, you know, I had my third task. So I want to do this P thing. I want to find perturbations. One interesting experimental design question would be, like, how can I now change my protocol space here to make these things that is now currently still missed? So can I come up with a computer design type of thing to maybe add this or that morphogen to go in this direction? I'll try to answer this a bit later, but I think so this vision of taking our models, not just to describe something a posteriori, which is sort of often what we may be able to do, right? Our training partner has a fantastic idea, we do analysis, we do something talk. You know, then helping us design the next question, I think is kind of really exciting. All right, so just to summarize, the first part, we built this reference atlas by essentially, you know, autoencoder or sort of more fancy type nowadays, transformer type of integration of these profiles. That's a medical impact. So Maria said there could be also some medical report here. So I just had this one slide. Rest assured that this is relevant. This is from a nice review from Sarah and Aviv some time ago, where, you know, even at the time, there's like a lot of data sets with cell that are being done now. And I think this is sort of a nice sketch of what, and particularly, you know, people that really were thinking what this can be used for example, for screening, which really kind of makes sense, right? If these assays get cheaper and cheaper, you know, you can potentially, I think, so we can potentially use this together with pharma to do now some organoid drug perturbation screen to see what type of plasticity or so is changing by perturbing the drug. So what you need to do for all of that is now to take your new OLAP generated data set and not just sort of analyze it by yourself with like a reclustering, reannotation, and so on, but I think now projecting this on top of the atlas, right? That's an interesting machine learning question. And again, like, how do you make it fit on top of it so that there's not like, there's no overlap or something? And we always know, like, if we have high dimension data sets, there's always going to be some type of big set of covariates that make it look different. You know, if I had someone who maybe earlier looked at some type of FACS data sets and so on, so even across different days, you know, in the same lab, these things sort of seem to diverge, let alone, you know, across different labs. So this data correction is a big thing here. And I think this metaphor that we did in this perspective, or really with Raoul some time ago, was that, you know, we used to mapping stuff. Like, I think, has any one of you ever assembled a genome for himself? Not really, right? Because, like, luckily we're standing on the shoulders of giants. Someone did it for us. We just map on top of it, right? There's sometimes some virus or sort of some maybe strange models people, but in this case, more classical. But I think it's similar here. You know, we might have now this modality or maybe a bunch of others. I think there's quite some excitement here. I heard with also spatial proteomics or maybe time information or spatial information. Essentially, once you have such a reference atlas, which could be all kinds of modalities, sure, you take a Newman method on top. And I think this is a bit the idea of these sort of bigger settings nowadays that go beyond. But, you know, at the time we've been thinking a bit about mapping. We had this nice sort of artist illustration. Let's say this would be your background reference data set. Then, like in this Google Maps fashion, you see this, right? You map your new data set on top, and most of these map to sort of existing cell types. And then there's maybe a new one being spawned because this one doesn't fit to the existing ones, and that's maybe a new, some type of cancer cells or whatever, right? This actually works. Just one example. In this case, we took an atlas from the lung and sort of tried to map a few, sort of act as if we had sort of this general map of human variations, added lavage samples on top of it, and asked, you know, where do they fall in this atlas? In this case, we took them actually from COVID samples with different severity. And what we see when we map these on top, we see that quite a few sort of those new samples seem to be falling in different regions. And because we have an annotation, we can look up which regions these are. So this one up there would be, I think, some CD4 or CD8 positive T cells, whereas this thing here seems to be sort of splitting up by severity. So the mapping just automatically puts disease grade in a different location. So there seems to be something in there. So what cells are these? Okay, these are macrophages. Let's zoom in. What's happening here? And then you can see actually that the severe ones were mapped here in the monocyte. I think monocyte-associated macrophages. This was tissue-resident macrophages, something like that. This was known before, but in this case, you sort of see it automatically by just mapping on top of the macros. I think these would be use cases that are nowadays quite commonly done. Yeah, we've been pushing then these type of mapping things a bit more, and I just have this one example that I wanted to show. There's been so often, we have a use case, and then there's some new methods being done. But interesting, in this case, there's this technique called optimal transport. Okay, so this optimal transport is a methodology. Let me just show you here that essentially takes some distribution and wants it to map to another distribution in such a way that you need to just minimally change things. Has been sort of a mathematical discipline for quite some time because it's sort of very nice to do math on. Essentially, it's maybe a bit simplified. But at some point, they discover the single cell field, and then now suddenly a lot of things happen. So in this case, you want to sort of minimize the length of these errors. Okay, that's the idea of the method. And we luckily paired up with this guy called Marco Couturi, a guy now at Apple, who has been in Paris, who has been pushing these methods to really scale up. So in this paper, we came together and sort of made a scalable version of that, and could now really do this for large-scale single-cell assays. And if you can now match distributions without, or maybe also with putting in priors, you can put in priors, but without, you could sort of say, hey, let's say I have a developmental image or single-cell RNA-seq or whatever observation of a temporal process. Can I try to interpolate these, and can I see, what would be sort of my intermediate thing? Can I see what's missing when I sort of morph this on top of the other, right? And that's one thing that we did here. For example, in this case, we had some sort of embryo development, and you could see that across those different time points here, I was just highlighting the heart cells that were sort of mapping up there, and that you could sort of see which factors drive them, because you do this mapping on a gene-by-gene basis, so you can really connect those time points into like a spatial temporal movie type of thing. So I think for these assays where you also want to combine time points, I think often, for whatever reason, we sometimes miss something between, maybe want to interpret other time points. I think that it's often quite robust way. Of course, nowadays there's also other ways, but I kind of like this approach. One other example we had here, we had one assay where you have RNA and protein measured, surface proteins called CytSeq. Okay, so you have these two measurements per cell, but dissociated. And here we had a spatial atlas. And the question was like, how can we now sort of enrich this spatial transcriptomics one, which typically is targeted, with also this protein information? And so it's again a mapping question. So I think a lot of these things that we do in some sense are putting something on top of another. And that was one of the methods that we did here. So to use this reference atlas is essentially, you would map them on top of this by essentially leveraging these representations. So that's been basically encoder-based type of things. But is this already like the model that we were promising? And now look like this, boom. So this would be this is a picture that you might have seen. This was CZI at the time when they were still working with the world, the days before Trump, sorry. So there, this vision of having the cell that sort of at some point gets digital is there. And this virtual cell idea, I think, is a nice one. And I think it's an old question. So to those old timers of you who have been around the age of systems biology, do you guys remember that we wanted to at some point build a whole cell? There were these first papers around. I'm sure, Rafael, you've been around with these things. So you like this graphs and stuff. I didn't say old timer.
I appreciate that.
Maybe I didn't. But I love this idea. Like when I came sort of very naively into the field like ages ago, I said, oh yeah, now we have the genome, right? We know how metabolism works. Let's just piece all of it together, and we build this big model bottom up that explains everything in a cell. And there was like a lot of, this was the type of predictions that were working out from these models. So you can see still an old MATLAB type of plot. This was not very predictive. And people even talked about virtual cell biology like more than a decade ago. This case was this minimal bacteria or something like that. But I think what's safe to say is that these models, not just not delivered, but I would say really completely failed, is super sobering up, right? There was so much effort in the mathematical side driven, but like nobody was using any of that, most probably because they sort of just didn't predict anything real. Like, there was no experiment designed to be able to done with this, right? I mean, just it was too rough an approximation, probably. But then, people took it up again. Demis, for example, was talking about this bottom-up AlphaFold sort of protein by protein type of cell. So this is sort of connection to your world to some extent, right, Bruno? But also, colleagues from CZI sort of pushing about this AI-based virtual cell, and there's not many others. So I think this vision is cool. And you have one of these colleagues, Charlotte, here, with whom all of us sort of put together this virtual cell perspective also. But I think this idea of learning and embedding, you know, this I drew before the cube, it's sort of the same thing, where you just know where your cells are, so this reference, but then you maybe also see where they would go and how to sort of change things. This is, I think, a very nice vision. Of course, not clear if we get there at all, but I think some approaches are for that. And I think what was maybe one of the Senate papers to go there is this next step in deep learning. You know, deep learning has been sort of, let's homogenize to some sense architectures, but now let's maybe even just train one model, not just for data set, a new one. Let's just train one big model, and that maybe sort of helps us. And I think some disciplines doing this, for example, in computational pathology, this is the way to go nowadays. So, you know, I think maybe say even just in computer vision, right? Like you don't make your own filters anymore. You just train on a lot of images. You take something pre-trained and you just take those. So the idea is take a data set, do some type of self-supervised pre-training. It could be these sort of more classical things, but of course, de facto also nowadays will be some sort of transformer-based representation. And this one doesn't have any questions asked on top of it. Just train it once on very big data. And once you do this, and then of course, that's where people brag, I have the biggest model and so on. And it's maybe not always needed yet, but then you can do some type of downstream things such as annotation and so on. And I think this idea is there. First one was by Christina here, this Nicheformer paper, and then a whole bunch of follow-ups. But, and this is important, also here, you know, I said, hey, this is the next thing that should maybe do the virtual cell. There was a lot of sobering up. There were a few other papers, but this was maybe the one that was really noticed the most by Wolfgang Huber and colleagues that said, you know, even for some very simple perturbation predictions, in this case, I think, I don't know exactly, I think it was some sort of genetic perturbation, even just, you know, linear baselines are a good job to do. And if some of you do sort of something cross-cell line learning, just taking the average prediction of a compound often is so hard to beat because there's so much noise in there, right? So that's why, you know, this sobering up. And, you know, there are some cases where you can do more. We just brought out this thing which is called Nicheformer, which is now a model that also adds spatial context. And, you know, this one, in my opinion, is so simple because the data sets were not so large. In this case, we actually take some spatial question. We ask, if you give me the RNA-seq profile of a cell, can you predict what its local neighborhood would be? Or can you maybe localize it in the brain, which regions, for example, the cell has been taken over? And interestingly, you know, even if it's an immune cell, we can tell you which brain region the immune cell came from, which at least for immune cells, I wouldn't expect so much, right? Because, you know, they have been talking to the neighborhood, that's why you can reconstruct it. And that's too hard a task that the linear method can do well. So in these cases, you know, we actually need these more complex models, I guess. So this been around. Now I want to show you something that we just very recently brought out, and we're really looking forward to your feedback on that. It's a bit more technical, but I know at least for Maria, maybe it could be nice. This is work by Moshtaba. And I think the current paradigm pretty much bases on this large language model type of approach, right? So essentially, we have a lot of data, but essentially then what we want to do is we sort of ask some question on the decoded data again. Typically, we mask something, we leave out a few gene expressions, and leave out like a spatial context, and I want to fill up that. This masked language models type of thing is the way to go. But people question is, and recently was at a very nice retreat that I literally got invited to. Is we're doing a high-level AI colleagues retreat somewhere in southern France in September. Do you want to come? Yeah, you know, southern France. And then this guy was there, Jan LeCun, you might have heard of him, one of the founding fathers of modern deep learning. He was sitting in the jacuzzi and discussing all of that. It was really nice. And his point was, and I really remember this, and you know, he's been sort of writing about this quite a bit. The way that we project everything down to language is maybe not the way to go to build these complex models, because you know, you lose concentrations, quantities, and so on. So maybe we need more to augment them. I don't think the vote is fully out. You know, there's sort of combined models that are very powerful in this. But his idea is, you know, let's do something more complex. And he sort of, one of his things is that, let's say, let's not go in reconstruction, because we know if you want to do gene reconstruction, these decoders are often very bad. So in our case, for sure, this is a problem. But his idea is, let's do this just in the encoder space. Okay, so just in the latent space without decoding. One of his things is called JEPA. We and others have been trying this bit for single cell, not yet so strong, but you know, in computer vision, there seem to be some promising approaches. Not fully scaled, but so what we propose is sort of a little bit in this direction, but doing something in the latent space. Essentially, let me maybe show you on the next slide a bit more. So what we call this SE concepts, we want to do some contrastive learning. Contrastive means of comparing things, not just reconstruction, so a different loss function. And it sounds a bit technical, but I think it's quite straightforward to understand. So please bear with me. So what we want to do, we want to learn better cell embeddings. You know, that's the task of such a foundation model. So we started our whole transcriptome samples, And now we just take sort of two distinct views. In this case, we would just take two different gene panels, such as what you would do if you do a spatial transcriptome assay. You have two different panels, and then you ask, hey, are these somehow be related to each other? So what we do, for this sort of embedding, we first limit the cells to this gene panel now. And for that, essentially, what you would do, you sort of encode them in a way that you can put them into a transformer. The typical encoding here, you do some sort of rank-based encoding together with some positional encoding. With technicality, you can play around with these. We've been playing sort of hyperparameter screening these a lot. This seems to be working the best for us at the moment. And then you put them in the transformer, right? And then you get the cell views that are sort of then learned from this. So just sort of stack a few of these typical transformer layers. Again, doesn't super exactly matter what it means. It's just like a high dimension parametric transformation that you can learn from the data. But now it's sort of the loss is important. Do you see that? Again, very proud of my animation skills this morning. Yesterday evening. So what we will be doing here is now we have our embeddings, and now we need to write down the loss. So the loss is up here. So let's say we have the embeddings from this panel, the embeddings from this panel. Now we can correlate them. And the question is, we want to have the embedding so robust that you can't tell whether it's from the embedding 1 or embedding 2. So you want to sort of make this as similar as possible to this one. So typical contrastive loss, there's various ways, it's called info-NC as one of the things. But this is sort of how this works. And now let me show you that. So the gist, you want to learn an embedding of the cells without reconstruction, right? So that whenever you take subviews, you cannot distinguish them. Okay. In this case, you need to be as informed as possible about the system. So you learn the biology. And now we ask different things in order to show you that this thing works. We typically do some downstream evaluations. The most common one that's done is just cell type annotation, a very basic one where often linear models do a good job. So I show you here the performance of the thing. We call it SE concept, which stands for contrastive cell pre-training or something. And here you see sort of these simpler models, also our earlier ones, and they do a good job in terms of two different metrics, but you see this is high, so that's good, okay? But interestingly, and here now you see that these foundation models are not so good. This is just training on everything, but you can also, instead of what's called zero shot, you can also sort of do it to a particular domain. In this case, maybe learn a PCA, or just do logistic regression. This is sort of state-of-the-art for annotation, logistic regression, okay, as basic as that. And this does much better than the zero-shot versions. But we can also fine-tune this model on the data, and then also outperforms this one, and this does it sort of quite systematically. And what's nice, in contrast to these things, this of course learns a co-embedding, and you see that sort of the query is nicely embedded into the reference. So the cell embedding seems to be visually working, but also in terms of numbers, and this is sort of consistent across other samples. So we train on healthy NMS patients. So we have a healthy reference, and then we query patients, which is sort of a harder task because there's a shift in brain cells. And you see again that this SE concept and the fine-tuned version are sort of outperforming this. Similar also for an aging atlas of skeletal data. So just sort of to summarize this thing, you sort of keep doing this contrastive learning. Contrastive learning is quite an expensive technique, but it's safe to say that in our case, the models are far from being even close to as big as in these language models, also in the vision transformers. This, there's some people bragging that they throw a whole bunch of parameters in there. This is not super large, and we can still robustly train. And you see that we sort of start outperforming all these sort of existing ones with this different loss. And very deliberately, we sort of put this as a loss paper type of out there, because you can take your own favorite method now. You can just sort of add, instead of reconstruction loss, you can ask this contrastive loss, and just to see if this helps you. And what's nice, because it sort of learns this panel, you can also ask, hey, what's mostly contributing to it? What's the attention sort of going in? And you sort of see that actually the differential top DGs in the reference sort of very nicely then be mapped more or less to the ones in the query. So these things are really being put together by this contrastive approach. So this is for annotation. You can actually do this also for cross-technology transfer. Because often one technology gives you maybe a different view of the data. I mean, this is sort of a reason why you do. Like if it gives you exactly the same data, why would you do the new technology, right? In your case, it could be maybe temporal dynamics. It could be like another set of proteins in spatial proteomics or so, right? Typically, you always have some targeted assays. So what we do here, we have a pseudo-xenium panel. Like xenium is a targeted spatial technique. In this case, we just take a panel, but apply it still to this data set to be sort of without domain shifts too much as a first start. And just want to compare now, we start with a reference and we want to transfer to a new set of donors. And what you see here is, so this would be the visualization in the UMAP of sort of mapping the xenium panel now onto the whole reference. You see it nicely intermingles, and you see the different cell types here. And interestingly, with these sort of previous techniques, interestingly also our Nicheformer, which is kind of bad, they don't integrate as well, or maybe even not. Okay, so it's really two different classes, and this sort of nicely does because it's been trained, to sort of make them forget their identity. Well, only here, but then it sort of seems to be generalizing. And this is also the quantification of how well sort of transfer performance in cell annotation works. So really, these two are outperforming many of the previous techniques, and we can quantify this again, and you can also see sort of in terms of accuracy. So just to make this point, this I think really gave us this robustness of technology transfer. So let's really try this now on a real panel. In this case, okay, take things on a seek, you want to map it to a spatial setting. And it's not as trivial as it sounds, because there's really these systematic domain shifts of the way these things have been measured. So this is what you get here. Again, zero shot versus domain adapted. And you see again, and just showing you a bit numbers here, but you see that there's really quite some strong gaps that you get versus sort of previous techniques, which was for me really promising. For example, there's this vision I've been talking with the guys from Allen Brain, where they build this Murphy atlas, and they want to sort of optimize maybe first the panels, because it's not so clear which panels to choose, but then also sort of how to really enrich all this spatial information with these previous big single cell RNA-seq atlases. And often these things don't go so well together. And this could be the way to go. So if you have that, could you maybe then fill up these targeted panels with the other type of transcript information that you get from the dissociated ones? So this is the idea here. So you have your single cell RNA-seq reference. Could you use this to impute the spatial assay? And can you first map it into it? So it's sort of this back and forth type of thing to do. Let's try to see how this works. And again, I'm showing you a bit numbers how this works, but there's a few methods that sort of try to do this before, and often just sort of very simple normalization by counts quite strongly outperforms both the foundation model settings as well as the other ones. And this sort of holds across a whole bunch of different data sets. So quite exciting to see that this does a good job. So I think we could use this, and we have some figures in the paper how you can use it also for panel design. The last point I want to make is, or second to last, is that we have these atlases. I showed you the lung atlas before. There, the embedding has been quite carefully trained through a lot of different sort of manual intervention steps and so on, because it sort of resolves all the cell type clusters. So sometimes you don't want to come up with a new projection, but you want to sort of take the old projection and sort of push your spatial samples on top of that. So this is what we try to do here. So let's say we want to fix the embedding here, which would be the dissociated embedding, and we want to learn a new spatial embedding so that it fits to the dissociated ones. Okay, so we keep part of the thing frozen. And because we have a flexible loss, we can do this. And this is something you can't sort of so easily formulate in the masked language model setting. And in this case, you can see that we have some sort of annotated airway epithelial cells here, and we predict them sort of very nicely to fit in the same region. And we can sort of see that this cell type label transfer, in this case from human lung cell atlas to a spatial human samples, is working well. And I showed you this architectural surgery technique before. This was this sort of Nicheformer mapping picture type of thing before. This is sort of what we've been using so far for label transfer, and they seem to be performing quite a bit better. So we're very, very encouraged by now trying this out. Last point, can you use this also for integration? So can we, for example, take in this case, so we have both. I think we had liver samples with some metastasis on top of that, and we have sort of both RNA, nuclear, and slide seeking. We wanted to put them sort of in a joint map, and you can see that the sort of technologies to quite some extent were overlapping, and we could do the same thing also in ovarian cancer, where you again see sort of some, I would say to some extent, robust technology overlap. Question is again, how can you do it previously? And then I'm done with my benchmarks. So you can come up with some type of methods again for the zero shot, and you could see here, there's a whole bunch of numbers, but maybe the total one here for the cancer, as well as the total one, or maybe you can look here, are sort of really doing a better job than the previous methods. Actually quite a bit of a jump. And more so if you do it sort of on the fine-tuned one, so de facto versions of this one, which will be sort of condition variational autoencoder, are the way to go. And I think there's a quite significant jump up here. So I was quite encouraged to now use it for cross technology integration. Last point on this thing that I want to make is, so now we come up with these more robust embeddings. Is this the way to go, or should we just go to language after all? So there has been 2 or 3 papers coming up with the idea that and we discussed it briefly at lunch, should we just take a big language model, where the information is in there, and we now build agents on top of it. Let's maybe So for example, you know, I come up with these problems, talk with colleagues from OpenAI, and then they said, yeah, sounds like an interesting task. Maybe we can add it to our big model. So you know, they just have this one model, and everything is added into this one, right? Computer vision is the same. Like you have this computer joint sort of vision language models. Is there maybe at some point a joint language biology model? You know? And there's one very naive, initially I thought, approach from David's lab from Yale. He calls it cell to sentence. He literally writes up sentences of where the highly expressed genes are in a cell type and writes up a sentence and then puts in the language model. Okay. I'm like, it must be super naive. But turns out that seems to be working not so badly. There's a sort of related paper. This is sort of more alignment type of thing, so a bit more clippy type of paper from Christoph's lab in Vienna called Cell Whisperer. And because you know I didn't believe this, so we played around ourselves a bit with that. So we call this now Cell Hermes. This was work by Gao. We changed from the lab, but essentially what we now do is we have a bit of modification of this idea. I thought maybe some quite clever modification, but this is in the end. It's we fine-tune a pre-trained LLM with question-answer pairs that we get from data sets. We sort of co-embed the data into that. And what's kind of cool is that you can, with this sort of, you get also sort of this very accessible way how to now interact with your data. So for example, these would be sort of 3 different examples that we show in that preprint, and I won't say more to that. But you know, you can sort of reuse this as encoding of different gene embeddings, and you can use these embeddings based now on the language part, which is astonishingly good. And we should sort of benchmark this a bit. You can use it for some type of predictions, and you can also sort of ask the model to explain what you see in the data sets. And it's astonishingly not stupid. So we should not underestimate the power of these really large-scale models to maybe search things that we don't expect. Of course, it's very hard to delineate what's actually signaled in there versus something that has been sort of seen and read in another paper that you might have been written, and we sort of catch it up. So like doing this really properly is not so clear. But there's so much of these agent type of papers coming up with it. I think, I mean, many of us are sort of playing around with it, and I think it's interesting where things go. Just sort of to summarize this part here, so I think we can do these robust cell embeddings, and potentially at some point we go a bit beyond this. So in the last 10 minutes or so, if I may, I would now take a step back and sort of talk about. I have another thing. I have something about other modalities. So I heard there's some people interested in proteins. So I have just like 3 slides on that. So we've been thinking about like could be maybe not just like a bad RNA. You know, cells in this very naive picture from us computation, they're a bit more than just a bag of RNAs, right? So proteomics at our place is a big thing. Matthias Mann in particular has been pushing these methods. And I think we asked, like, can we really go single cell proteome level, right? And I think it's a kind of nice vignette to briefly show. So, you know, so people have been upscaling of novel proteomics, and single cell proteomics is also capturing now quite a few things. But what you see in this paper is that it's still sort of very manual. So, you know, you can't produce hundreds of thousands of these single cell proteomics. But we tried a bit, and sort of we had this one collaboration with colleague Erin Schoof and Bo from Copenhagen, where we've been essentially trying to really now go into an in vivo situation, namely human bone marrow, then sort of really at scale isolate single cells, and then, you know, run them in this very sensitive mass spec to really do single cell full mass spec. And we were able to identify across more than 2,000 cells up to 3,000 proteins. So that's sort of the largest data set at the time to really quantify, not just in a label-based surface marker type of thing, but, you know, really sort of clearing things out type of proteomics. There's sort of other approaches where you laser capture microdissection type of things, take out a bit more mixture of things. In this case, it was really pure proteins, right? And this is then the UMAPs that you get from that. And you see, you know, it's not sort of as nicely resolved because these are things a bit more noisy. It's not as clear as these labeling-based techniques and things are seek, but you get your hematopoietic stem cells sort of rolling down the hill to these various lineages as you would expect in hematopoiesis. You can annotate them. And I think that's kind of nice. You can potentially now integrate, again, with these mapping techniques that I showed you before with other data sets. So we asked here, you know, I mean, our dream, of course, would be we want to have RNA and protein in the same cell to really model translation, like do all these systems logic type of things. Doesn't work yet. People working on it. But we have this assay called CytSeq, which gives you surface marker proteomics and RNA. So we could use this to couple it. So what we did a hematopoietic CytSeq data set and coupled this one with this joint latent space integration with the proteomics one. This was worked by Sabrina in the lab together with Benjamin. And then you get this joint latent space where you now not only have the RNA ones, but also the protein ones. You can sort of put them together. And that's kind of cool then, because then you can ask questions. And one question, for example, would be like, how much information is now in the protein versus the RNA? And you get sort of some sort of particular markers up that you wouldn't see in the RNA because of the dynamics and so on. And I think you can also build a model for that. And that's what I really like, because, you know, we want to build up some equation type of things here to maybe understand. And one thing we could say is, you know, if the mRNA goes up, obviously, you know, if it's a protein coding one, your protein will go up a bit after. So if you sort of plot those two concentrations, you have a few samples, you would see this type of dynamic behavior. If this thing is upregulated, if it's downregulated, it would sort of look a little bit like that. These are these typical examples that a guy here in the audience, Joelia, has been, I think, seeing for unspliced splice reads. And we've been sort of trying to generalize this a bit for this setting. And we can then learn some type of dynamics here. And I think this is kind of cool. We can then also try to associate how much the RNA tells us about the protein in these single cells. And you might know these bulk measurements, typically something like 30%, so it's not so much. It actually goes up quite a bit. If you go on the single cell level, then it's maybe 35%. And if you now include this type of lag in the model, it goes up to close above 50% or so. So, you know, this sort of information richness is kind of contained there. So that's something we really found nice. The second thing on other modalities I wanted to show is this morphology type of embedding. I put this in because we just chatted about you guys being interested in sort of morphometric type of measurements. And we've been putting some effort in. This was work from Niklas also together with Matthias's lab, who in this case just used in computer vision. It's very hard to beat these masking type of losses. So that's what we did there. So just learn the representation of the cell variation of now lots of images. And the nice is there's some sort of package with it. So you can really scale it to billions of cells. I think the engineering part is nice. What you get is now sort of similar type of view maps as you saw before, but now not variation because of RNA, but just because of sort of change in morphology. And maybe if you have sort of surface markers, you can add this as well. And you see, you know, there's some group that sort of had this shared morphology and so on. There's been, I think, Lukas Pertmans, no, maybe it was Prisca Liberali's lab, with Gabriele, maybe, where they sort of did this also morphometry of organoids and so on. So this thing has been done before, but what we do here is sort of at scale now do this, in this case, actually sort of superimpose with a Xenium image. So we can now look into particular clusters, and we find one cluster here, where we also now have the superimposed Xenium image. You can say, where's this heterogeneity coming from a spatial morphometric change in sort of the cell clusters? And you see, okay, there's actually a few sort of RNAs in the differential change. This was some ovarian cancer setting. So yeah, you can sort of put them together and computationally integrate them in the models. That's maybe a little summary of this part. And now the last thing I want to say is, of course, like as a vision, we now want to go towards perturbations, right? And I think there's some approaches to these perturbation settings. This is sort of from a perspective where, let's say, you have this rolling down the hill of what is landscape to become maybe in a severe disease. How do you want to invert it? Maybe you have your sort of virtual cell that tells you how you behave under perturbation, then you pick this drug to sort of revert back to the healthy case, right? So this is sort of that idea. But this does not really yet work. You know, I showed you, right? Linear models often doing enough. And I think the questions are not so hard. I mean, we spoke about the very strangely named virtual cell challenge from ARC, right? Which de facto was a perturbation challenge that were also, I think, kind of destroyed because maybe questions are put so clearly. So, but the number of data is actually getting so big that the questions we can ask to these is getting exciting. And I don't know what the scale is of the transcription factor dosage perturbation you guys do, but I guess it's also more than just one or two perturbations, right? So here's one data set we've been looking at. This is from a company. There's a few competing companies now that sort of do similar things. But what's kind of really cool, in a single experiment, generating more than 10 million transcriptomes, right, across more than 1,000 conditions. That's really cool. In this case, they took 12 PBC donors, looked at 90 cytokine treatments per donor ex vivo, and then sort of come up with this dictionary of cytokine responses. We're currently analyzing this, sort of also trying to build a shared dictionary for that. It's a really nice data set. And then, you know, CCI has been putting out a 100 million challenge. Tahoe has this 100 million cell one. There's more of these. And, you know, our tools work on that scale, so that's useful. But what can we do with that? And this is the last thing that I want to show, just going back to our organoid setting. So we came up with this technology, or with this sort of approach, which we call a Cellflow. This is work from Dominic, together with Jonas from Camp Lab at Roche, where, you know, you want to now take some of these large-scale data sets and try to learn a predictive model for sort of how these cells behave under perturbation change to then sort of impute new perturbation changes. For that, you need to sort of put these in a joint space. So there's a few challenges. First one, of course, like these things are typically not paired. Traditionally, if you learn diffusion flow matching models, you typically know how before and after looks like because you have a movie. In this case, we don't have a movie, so we need to pair them. We use this optimal transport type of technique. We want to have more complex perturbation settings because these experiments typically are not as simple as, just add protein one or not, but it's a dosage, it's combinations, so on. So for that, we have some type of attention mechanism to do so. And then we want to make realistic cell profiles, so this generation. And for that, we use this technique called flow matching, which is sort of a, let's say, more robust version of diffusion maps. This is the stuff that when you talk to your GPT model to draw you funny images of Italian brain rot or what your thing is, right? You can do with that. And this actually works nicely. So I don't show you that it works nicely. I just show you, because I don't want to go over time too much, how this works for organoids. So for organoids, you remember in the beginning I said I want to use this atlas to design now an experiment that really sort of does something meaningful. So in this case, there were a few data sets where people did already throw different morphogens on top, and we sort of put them all in a joint space. What exactly that means doesn't really matter, but we sort of unified a bit time points, concentrations, so on, into sort of one space where we can now play around with different protocols. And we can then leave out one data set and try to impute it based on the other data sets, and we see that the Cellflow does a good job, okay? So that gave us some hope that we can go completely out of distribution. So for this, that's what we call this a virtual organoid screen. So we take now a bunch of these base protocols, add different modulators in different combinations and different time points, and thereby completely in silico in this space generate now 23,000 different protocols. Not something that you want to do in your lab, right? Like setting up a new protocol 23,000 times, maybe not. But because it's computational, we can generate cells now from these ones. We generated 70 million virtual cells of these various protocols, and we group them, and you can see what shared patterns in there. And yeah, you get something. But now you come into a really exciting problem. So this is now a virtual experiment. Like, what do you do with that? Yeah, you can believe it or not, but what's exciting in this? So now it's an experimental design. Like, what's actually meaningful? What would be a good recommendation or experimental partner to follow up? So we try to quantify a bit how similar so each of these dots is now a protocol. And we quantify how similar or distant they are to the, in this case, primary reference, so the human reference. This one would be sort of very similar to the fetal development, not very dissimilar. So we kind of prefer similar. And here, how close or distant it is to the training data. So this stuff we kind of know, but this region here is something that is new, but similar to the human situation. So then you can come up with a few and you can sort of look at them and see what they do. And these things are now to follow up. So I think for experimental design, there's this very interesting question. Once we can actually have a model that does something under perturbation, which spaces of this parameter combination that we can come up with should be explored? Maybe something where uncertainty is very high, maybe something where what we want to generate is high. So there's interesting methods to be gleaned upon. So to summarize this part, I think we can do this data-driven type of modeling. We do flow matching. There's many other approaches around. I'm sure you have something related here as well, but I think we can extrapolate these cellular response profiles. And if I may, just one more or two more slides. Sorry. We can actually show that this works to something. So this is the vision, right? So let's say you put the lab in the loop and then you sort of go around, right? Let's say if you really can build a robust model, you can do actually perturbations and you can do an experiment, you can sort of follow this up. This is sort of this big vision, this systems biology loop that, we proposed like field proposed 20 years ago, right? But this is actually a bit happening. So we had this one collaboration with a company, it just came out a few weeks ago, where the core model was super basic. So in this case, we just wanted to predict how a transcriptome signature upon a drug perturbation was actually mapping out. So in this case, we took a database called connectivity map, old thing from the broad, had a very simple, just multi-layer perceptron to predict what each compound sort of has in terms of efficacy for a particular question they had. And then we saw that this model was doing better than simpler models, so that's good, and we had some type of prediction. But the key novelty in this paper was that we threw a lot of money and data at it and sort of updated it based on this prediction and sort of had a way how to update our signature that we originally get. So we had sort of this recursive learning loop. I don't even want to call it a full reinforcement learning, so just call it active learning. But this actually worked out and we get some sort of better signatures, and this was now nicely followed up, and we have a few examples in the paper that I encourage you to check out. So with this, I just want to wrap. So I think this idea of these large scale models, and I don't even want to call them foundation models, but I think just coming around with all these data sets. We discussed about this, but also compute resources, there are challenges. how to evaluate this. I think these benchmarks really make sure that whatever we do is meaningful, is not clear. This is from a perspective we recently wrote. Some ethical considerations, but then also like just taking time and making sure these things work. So I think particular benchmarking that stuff is so important. We have this open problems consortium, contributor things there. Let's make sure it is good that ARC and others do these challenges, but if they don't work, we need to make them better. But I think this is the only way how we can convince our experimental partners to really take up something like that. With this, I really thank you for your patience and attention. This is my lab. We also have some mountains close by, but this is actually a retreat really in the mountains. This is also how the weather in Munich is always like. And I appreciate your attention.